right. Okay, so during ParaFishControl, we in the Czech Republic worked on a parasite in carp, and this parasite is called Sphaerospora molnari. And as part of our research, we also investigated candidates for developing vaccines. So who is Sphaerospora molnari? Not all of you may know this parasite. So it's a myxozoan, the same as Enteromyxum lay or the famous Tetracapsuloides bryosalmone. It causes serious pathological changes in especially the gills, but also the skin of common carp. You can see this here in this image. Everything that's labeled blue is parasite, and you can imagine that the fish can hardly breathe under such conditions. So the parasite is also found on the good research model because it actually develops these developmental stages, proliferative stages in the blood that we can find prior to the spore formation in the gills. And we were hoping by working on the proliferation model from Sphaerospora that we would have a model with transferability to other myxozoan parasites. So what candidates did we look at during ParaFishControl to help us design a vaccine in the future? So we took two approaches. One is the obvious one, that we would look at antigenic surface proteins that cause immunity and that will cause protection to a reinfection in fish. And then we also looked at another group of protein that's important in it plays a central role in pathogenicity and in host-parasite interaction, and that is proteases and their inhibitors. But let's look at antigens and their immunity first. So our first question was obviously, are there any specific antibodies produced to Sphaerospora molnari antigens? So once we developed our in vivo model of infection of the parasite in the carp, we could look at this. And you can see here that during the first 14 days of infection, on the left graph here, you can see the parasite is hiding out somewhere. We still don't know where this is, but it later shows up as a peak of infection here around 42 days, and then there is only smaller numbers of parasites to be found. And this matches nicely the lymphocyte profile that we find in these infected fish, and even better, the profile of B cells. B cells are involved in antibody responses, so that was a good sign. And in fact, when we run parasite protein extracts on a protein gel, you can see if we plot this with sera from different days of infection, the sera are able to recognize certain proteins of the parasite. This one down here, that's about 15 or 16 kilodalton, is one of our major interests. But you can see later on in the infection, we don't just find one, we find several proteins, and this one actually at an even later stage disappears. But okay, the candidate is good because it mirrors this 15 kilodalton protein mirrors the infection and the IgM B cell reaction to it. So yes, the answer to this is yes, we have a specific antibody production to the parasite. So we can use this as a starting point for trying to find those molecules. So do these antibodies have killing capacity was kind of our next question. And here we ended up with the same problem as on the previous gel that you saw. So yes, the antibodies do have killing capacities. And you can see here, these are 3 different lots of parasites that isolated at different times during the infection. And you can see that the antibodies in the same serum are sometimes able to kill all the parasites, whereas in other parasite lots, they kill only about 20%. What does that tell us? Yes, the parasite has obviously more than one protein of interest and more than one protein to make the carp react to it and produce specific antibodies. So, okay, but another question was to know whether the antibodies are actually important to the infection. So could carp also survive if it wouldn't produce antibodies? For this, we developed a model of immunosuppression. You can see here, this is a normal infection, and this is a carp group that was immunosuppressed and infected. Our immunosuppression reduces or eliminates basically the pool of B cells, so they cannot make antibodies, and this has really a quite substantial effect on the fish. So the parasite feeds on erythrocytes. So here you see the number of erythrocytes, and in a normal low-level infection, you would see a slight decrease in erythrocytes numbers compared with the control. But if you do the immunosuppression, you can see that the parasite takes over, feeds, well, basically consumes all the erythrocytes, and without the specific immune response, the parasite would actually kill the host. So immunity is important, and those proteins are important. So can we use them to immunize carp against Sphaerospora molnari? And that worked really nicely. So you have the same two bars of parasite numbers here again. And then if you look at, if we inject the fish previously to the infection with the parasite with sera from immunized fish with inactivated parasite, or we give it like a second dose of infection, they're actually completely protected, and there's none to hardly any parasites in these fish. So that's good, yes. Both the parasite extracts and the immune sera can provide protection. Now, the only step left, this is the lengthy step, of course, is the identification of these antigenic proteins of Sphaerospora molnari. It's been quite tricky, but we've learned a lot. So the obvious method for doing this would obviously be mass spec. Now we've learned that we have as much fish proteins on our gels as we have parasite proteins, and things are difficult to identify when your hemoglobin subunit is the same size as your target protein, and similar things. So we've tried also things such as immunoprecipitation, which we've come up with some candidates. And finally, I think what is helping best is the parasite membrane proteome. But we're slow on this because we need 500 million parasites to have enough material to do the proteome of the membrane. And that of each parasite lot with a different protein surface. So we're on it, we're close, and we need to work together because we found out that in infections with Enteromyxum leyi, for example, we also find a similar protein as a similar kilodalton size protein in infections in the gilt-head sea bream. So we will compare and hope to find something that is of value to more than one species. Okay, and now just very quickly, the second approach we took are the proteases and inhibitors. We first looked at the protease household of the blood stages of our parasite. You can see we have a large number of proteases. The important bit is here that we have about 38% of cysteine proteases, which are not usually that common in other parasites of the Myxozoan group or in free-living cnidarians. What we were looking for now in this pool and in the whole pool of proteases and inhibitors were candidates that have a signal peptide, because you do want to have a signal peptide to make sure that this protein is actually used outside the cell as an intermediate for interacting with the host cells. So the most highly expressed protein we could find is a cathepsin L of the parasite, and we thought that this may be a feeding proteases, feeding protease, so proteolytic enzymes that the parasite uses for feeding. What you can see in the gels of the recombinants is that it's in the total and in the pelleted phase, but not in the soluble phase. So the protein is not soluble. And then when we tried to activate it, we also saw that at normal pH, we cannot really get an activation, we have to have a pH of 5 or better 4.5 to get the protease activated. And that kind of indicates, together with the fact that it's not in the soluble phase here, that it is likely inside lysosomal compartments. That would make sense if it's feeding on it. So we don't know from that. Here you see some images of the parasite feeding on the red blood cells and on the transport of proteins from the red blood cells to the parasite. So yeah, if it would be feeding, it would use this in a lysosomal compartment, and then we don't know if it's going to work as a vaccine. But it may be used also externally, so we just have a vaccination trial on this ongoing, the first one.
Sorry,
and the last candidate
left, sorry.
Yeah, okay. So the last candidate we looked at is a cysteine protease inhibitor that's active in the gills, where we haven't gone that far with it. I'll just tell you, we found that it inhibits cathepsin L, and that we've made an antibody from the recombinants, and we can visualize it, but we don't know yet what it does in the gills because we haven't had time to look at the gills yet. We only know about the blood stages. So we'll get there. Just in conclusion, a carp acquires specific immunity, and I think that's the right way to go to look for antigens that we can use, and that we can use their immunogenic potential. The antibodies protect the fish from infection and can kill the parasite. The identification of the antigens is difficult, but we are on it. And we likely need to use a combination vaccine to be able to combat the immune evasion strategy of the parasite. We expect high effectivity and hopefully transfer possibility of this research to other myxozoan parasites. And the protease and inhibitor research is still more in initial stages, but it has improved our understanding of the myxozoan host exploitation quite a lot. Yeah, and I would just like to thank ParaFishControl for making all this possible and believing in our carp model, which I think has advanced things quite a lot. Yeah, and that's me.
Great, thanks very much, Astrid.